Hello, we will be presenting our research on the potential role of post-translational modifications in SARS-like coronaviruses as an infectious driver. My name is Daniel Morales.
My name is Jessica Gonzalez, and our faculty advisor is Dr. Jessica Siltberg-Lieberlis.
SARS-CoV-2, the virus that causes COVID-19, has infected over 110 million people worldwide. SARS-CoV-2 isn't the first coronavirus to infect humans. In fact, it is the third emergent coronavirus since the turn of the century. However, of the three recent coronavirus outbreaks, SARS-2 has had the highest infectivity rate. Post-translational modifications, or PTMs, are molecular extensions added onto certain amino acids on proteins. These modifications help regulate protein function. Viral hijacking of certain PTMs have been linked to increased infectivity and pathogenesis. Our objective was to investigate the relationship between PTMs and increased infectivity in coronaviruses.
We built protein families for all coronavirus proteins. Post-translational modifications were predicted and highly conserved sites were determined. PTMs were integrated with our MSA in trees in order to put structural and evolutionary context to the data. Statistical testing was done using Shapiro-Wilks and Mann-Whitney U-tests. Using NSP12 as a representative phylogeny for the proteome, 3 clades were identified: the SARS clade that includes both SARS-1 and SARS-2, the MERS clade, and the OC43 clade. The coronaviruses are colored based on their host: red for human, blue for bats, and black for other mammals. SARS-2 is denoted by a gold star in the red SARS clade. Using experimental data about the subcellular localization of the proteins within infected human cells, in tandem with our PTM predictions, we were able to observe significant differences in the number of PTM sites per protein between clades. Overall, SARS clade has more PTM sites than MERS and OC43 clades. For example, Spike has more PTMs in the SARS clade with 18 PTMs as compared to 7 in MERS and 5 in OC43 clades.
Here we can see how the SARS clade has a wider distribution of PTM sites across all of the proteins compared to the MERS and OC43 clades. This means more viral proteins in the SARS clade may be modified than the other 2 clades. Furthermore, we found that the SARS clade had much greater diversity of PTM types than the MERS or OC43 clade. The SARS clade PTM distribution is composed of 12 different PTMs, while the MERS is composed of 7, and the OC43 clade is composed of 6. This chart shows an increase in the number of highly conserved clade-specific PTM sites in the SARS clade compared to the MERS and OC43 clades. It also identifies several PTMs uniquely specific to the SARS clade, such as s-palmitoylation. Let's dive into one of these modifications. S-palmitoylation is a key example of how these PTMs can drive infection by helping anchor the spike protein into the viral membrane. On the figure in red, we have highlighted the s-palmitoylated sites located in the transmembranous region of the viral capsid. On the bar chart, we see that the SARS clade has several clade-specific sites with almost full conservation. The box plot shows a statistically significant increase in the number of conserved s-palmitoylation sites in the SARS clade compared to the MERS and OC43 clade. Altogether, this suggests that increases in the s-palmitoylation sites in spike may increase viral entry and infectivity in the SARS clade.
We have found that the SARS clade has significantly more predicted post-translational modifications overall, and is more diversely modified than the MERS and OC43 clades. Diverse PTMs in viruses may enable them to infect a greater number of hosts and offer increased adaptability to a wide range of host environments. This large increase in PTM diversity in the SARS clade must be further investigated and may help elucidate the infectious nature of SARS-CoV-2. These PTM sites may serve as possible infectious drivers with rapid evolutionary rewiring. I'd like to say a special thank you to FERC for providing us the opportunity to talk about our research. We'd also like to thank Dr. Jessica Silberg-Lieberlis and Janelle Nunez Castilla for their mentorship and guidance throughout our project. Thank you, everyone. We will now be taking any questions from the audience.